Now we are going to discuss about the differences between the chemostat and the turbidostat. So far, we have discussed the dynamics of microbial growth in different culture systems or growth systems such as batch culture system, continuous culture system, synchronous culture system, and biphasic culture system. So here the chemostat and the turbidostat differences comes under the heading called as continuous culture system. That means these two types are going to be of continuous culture system. So what is a continuous culture system first? A continuous culture is essentially a flow system of constant volume to which medium is added continuously and from which continuous removal of any overflow can occur, and the microbial population can be maintained in the exponential growth phase at the constant biomass concentration for extended period in this continuous culture system. So as I told you, we are having the two types of continuous culture systems that are commonly in use, or one is the chemostat and the second one is the turbidostat. So let's have just a glance of what is the chemostat and the turbidostat, by which you will know about the differences between these two. Coming to the chemostat, this is a most common type of continuous culture device, and the chemostat permits control of both the population density and growth and the growth rate of the culture too. And this is going to be of mainly constructed so that the sterile medium is fed into the culture vessel at the same rate as medium containing microorganisms is removed. And two elements are going to be used in control of this chemostat. That is, one is the dilution rate and the second one is the concentration of limiting nutrient, for example an amino acid. Because of the presence of an limited nutrient, for example amino acid that I told you, the growth rate is determined by dilution rate in the growth chamber. And finally, the cell density depends on the concentration of limiting nutrient. And coming to the turbidostat, the second type of continuous culture system is this turbidostat, and it is going to be provided with a photocell. So where you can see this one photocell to measure the absorbance or turbidity of the culture in the growth vessel. So this is a growth vessel where you are going. So for the detailed explanation, you can go to the previous video. The flow rate of the medium through the vessel is going to be automatically regulated to maintain a predetermined turbidity or cell density. And this is going to be achieved by monitoring the biomass with this photoelectric cell and feeding the signal to jump, supplying the medium to the culture such that it is going to, this valve is going to be switched on and switched off if the biomass exceeds the set point, and is switched off if the biomass falls below the set point. So this is going to act as a controlling of the flow of the sterile medium into the growth chamber. So how it is going to be? So this whatever the signal that is going to be given by this one medium, the pump is going to be switched on if the biomass exceeds and the set points. So that means whatever the concentration that we are going to set, that is going to set. And if the biomass falls below the point, again it is going to be switched off. So like this, switch on and switch off, this turbidometry method is going to be useful in regulating the constant concentration or turbidity or cell density in the growth chamber. So by seeing the main four differences between these two, the first one is going to be the regulatory factor in the chemostat is going to be a nutrient present in the limiting quantities. Whereas here in the turbidometry, you can see the regulating factor is going to be the turbidity of the medium, which is going to be monitored by the photocell. And here in the chemostat, dilution rate remains constant, whereas here the turbidostat dilution rate is going to vary. And the limiting nutrient is going to be present. I told you, for example, we have taken an amino acid as a limiting factor, which is going to be deciding the dilution rate, or what is the thing, concentration of limiting nutrient. Such type of the limiting nutrient is going to be absent in the turbidometry or turbidostat. And chemostat is going to operate best at lower dilutions, whereas the turbidostat operates best at a high dilution rate. So this is the brief or overview of the differences between the chemostat and the turbidostat. Thank you.